So good morning or afternoon or evening, everyone, depending on where you are in the world. I am Tom Helton, as Brian mentioned, a former member of the Roth Lab, and now a co-founder and the chief operating officer of e-IDG. It's a focused research organization. I'm going to be talking along with Jeff DiBerto, who's also a co-founder from the Roth Lab. And is the current director of asset development at e-IDG. So kind of as a quick background to start with, people may ask, what is a focused research organization? FROs are kind of a new, FROs or pros, should we say, are kind of a new research effort. They are designed to have a 5-year limited time frame, and they're created really solely to solve scientific or technological challenges that are efficiently addressed in the current academic, industrial, or governmental organizational structures. They look to enable significant advances in areas requiring high levels of coordinated engineering or system building that academia really doesn't attempt, and also the outcome is to really benefit society broadly in ways that industry can't rapidly monetize or actually doesn't care to monetize. And finally, FROs, their primary aim is to produce well-defined tools and or technologies or key scientific data sets, refined processes, or resources that are dynamically designed to accelerate progress within a technological or scientific field. And essentially they are in this sweet spot between large-scale corporations, early startups, and academic researchers or government-funded academic research. So the next question is, all right, so we know what FROs is, so what is the kind of the creator, or what is Convergent Research, which is a member of the Schmidt Futures Network. So they are our parent company or our parent organization. They are a 501c3 nonprofit. Their primary goal is to incubate, find philanthropic donors for and launch FROs. And they also provide operational support for the FROs throughout their 5-year duration. And also, very importantly, is they're comprised of scientists and technical experts that have a wide-ranging combined expertise from academia to government to biotechnology. And I wanted to point out in particular, this was Tom Khalil, who was a scientific advisor for 2 presidents, both Clinton and Obama, and he was actually a very large champion of the EVE initiative. So now that we know what Convergent Research is and what FROs are, so who actually funds these endeavors? Well, currently there are anchor donors. The first and foremost is Schmidt Futures. This was funded Schmidt Futures was funded by Eric and Wendy Schmidt. I think most people know who Eric Schmidt is. He's the former Google CEO from 2001 to 2011, when Google was really kind of becoming a mainstay in worldwide online activity. And secondly is another large donor, is Kenneth Griffin. He's the founder and CEO of Citadel LLC and Citadel Securities, which is a multi-billion-dollar international hedge fund. And people may or may not know that we were actually mentioned recently in the press, and particularly the Forbes article that came out a couple of weeks ago, both describing why Ken Griffin and Eric Schmidt are spending millions of dollars on this new kind of FRO effort, really kind of showing that focused research organizations, here the caption is the aim is to provide useful information to the scientific community in areas that aren't currently being served by academia or business. And Eve and another one of our FRO partners is each has a pretty nice little paragraph description there. So please look that up if you are interested. And finally, so what is the current FRO portfolio? As of right now, there are 4 FROs supported by Convergent Research and the subsequent Schmidt Futures and Ken Griffin's group. One is e-11 Bio. They are looking to develop scalable technologies to pipeline and map the structural details of a mouse brain. Cultivarium, which is they work in non-traditional biological organisms or non-biological organisms. Helping use these diverse microorganisms for beneficial synthetic biology technologies. More recently, PTI, or Parallel Seq Technology Institute, came online just before us. They're looking to do single cell proteomics for the exploration of unprecedented scale and resolution for human disease, underpinnings of health and human disease. And finally, us as e-Bio aiming to identify approved pharmaceutical drugs, unintended or unknown molecular targets. So this is probably a little bit of a review for most people. This kind of gets at one of our core reasons for doing this is that, as many of you know, that drug discovery is really set up to fail. That currently more than 90% of all drugs that hit clinical trials for various fail for various reasons, including efficacy, safety, commercial or other unknown or unspecified factors. And that what goes along with that is that the cost of drug discovery increases every year, while the number of approved molecules to hit the market actually decreases. And this is also known as Arun's law, or the opposite of Moore's law, which really states that increases as production increases and costs decrease in relation to gains in production experience. This is kind of more so towards electronic circuits, but still applicable here. And finally, even post-drug approval, new side effects are still being continuously discovered for drugs that hit the market. So for drug discovery, really the ideal idea for drug discovery is that you have one drug and it hits one target or a very specified group of targets. As an example, this is a very well-known drug, is Lipitor or atorvastatin, the cholesterol managing drug. It's one of the most highly tolerated drugs on the market and has been for years without minimal or even manageable or even minimal side effects. And currently, about almost 25 million prescriptions are given annually for Lipitor, or it's off-patent generics. And while it was on patent, Lipitor itself generated almost over $14 billion in annual revenue for Pfizer, which at the time was 1/4 of their annual revenue. So this is kind of the ideal scenario. But really, what more so happens is you discover one drug, and it turns out that there are multiple targets, and you try and manage those multiple targets. In this case, this is one of the extremes, which most people know, at least from my generation, in the late 80s and early 90s, the fenfluramine, phentermine, or fen-phen, which was touted as a miracle weight loss drug. Turned out that after a couple of years on the market, that there are reports of up to 30% of patients taking this drug develop valvular heart disease. Ironically or serendipitously, in 2007, the Brian Roth lab discovered that it was a metabolite of fenfluramine, also known or called norfenfluramine, activated the 5-HT2B receptor in the valvular heart tissue, and led to abnormal tissue morphology. And finally, there's been more than $28 billion in damages paid out so far on this drug that was pulled from the market in the late 90s. So at this point, I'm going to kind of turn this over to Jeff to let him talk a little bit more about the technologies and so forth of what e-Bio is going to be taking on.
So you can advance. I think it's worth pointing out that this FRO, Eve, is really the brainchild of Brian, who was approached initially by Convergent Research for transformative project ideas in the realm of pharmacology. And so really what Eve is doing is sort of putting into expanded experimental practice this paper that was published collaboratively by Brian as well as Brian Choi-Kett's labs in Nature, where they performed in silico docking of more than 3,500 FDA-approved drugs against hundreds of receptor targets. They then followed up on these with 30 computationally predicted hits and tested them against a subset of these targets, finding that 23 of these target engagements do occur with, ligand-based, ligand-based predictions with potent functional interactions of less than 100 nanomolars. And even from what might be considered a relatively small data set, there's been a number of valuable findings. And so Eve will seek to experimentally test a much larger number of potential interactions between ligands and receptors. Go to the next slide, please. So drug discovery typically occurs through targeted screening at a receptor or a few receptors of interest, although in some cases FDA-approved drugs are found to be therapeutic through serendipity. And so while the inclusion of some known anti-targets such as the aforementioned 5-HT2B receptor may be screened against to rule out liability issues for safety, the known pharmacology of these drugs is usually incomplete and in some cases unknown entirely. So even when there's more thorough screens conducted, the data obtained from these are usually proprietary for and preserved by the industry that's conducting the screens. And so just from a public standpoint, and academic standpoint, there's a lot of valuable information that can be gained from obtaining a better understanding of FDA drug pharmacology, which itself may lead to future drug discovery to enhance efficacy and safety of currently available medications. Get to the next slide, please. And so the general approach here is simply to map what we were referring to as the pharmome. Here we will test a number of compounds against a large percentage of the druggable genome. If you go to the next slide, please. This will consist of about 1,100 protein targets. Just given our particular expertise, again, we both come from Brian Roth's lab, we will be starting with GPCRs. Part of this is for comfort, but also these are the most highly targeted receptor family by FDA-approved drugs. And in addition to simply identifying agonistic or antagonist actions of drugs of these receptors, or these receptors and others, in particular for the GPCRs, we also seek to expand upon some of the current information we have with regards to agonism, looking into signaling texture, which includes actions at different transducers relevant to concepts such as bias signaling. And we hope that at least in part these may shed some light on differences where chemically similar drugs diverge in their therapeutic safety or efficacy, and will help to inform more rational drug design in the future. Go to the next slide, please. And so our drug data set will consist of about 2,600 drugs. 16 of these are FDA-approved small molecules, including short peptides, as well as 800 of their known active active metabolites, which themselves can exert both therapeutic as well as deleterious effects. And we're also interested in trying to parse out the reasons why some drugs may have failed prior to making it to the clinic. And this will include a list of about 200 withdrawn drugs or those that failed prior to gaining FDA approval. Next slide, please. And, so we plan on conducting these assays in live cells. This is pretty much standard for the field of drug discovery at this point. And to the extent that it makes sense, use robotic screening. In order to facilitate the large number of targets, we are going to employ multiplex assay approaches such as those previously described by Shvir Kasori and others, both when he was at UCLA and now at Octave Bio. And then to continue in the spirit of the IDG, as Brian mentioned, we are interested in developing assays for orphan receptors at which presumably some of these FDA-approved compounds may act. And this may be useful for not only identifying perhaps currently unknown efficacy or safety factors and how certain drugs work, but also providing probe compounds for further study of these receptors. Next slide, please. And so the pharmacological benefits of mapping the pharmome will help to identify currently unknown off-target partners for existing drugs. And altogether, this data set should help improve predictive tools to avoid off-target side effects, thus increasing safety, as well as support the rational design of multi-target drugs. This is the concept of magic shotguns versus magic bullets. And we may have some serendipitous findings where it makes sense that currently FDA-approved drugs could be repurposed for other therapeutic indications, which would cut down on both time and expense necessary to develop a drug de novo. One of the hopes with our data set is that we will be able to provide unique, high quality training set for artificial intelligence for machine learning purposes, which itself may drive novel drug discovery efforts. So with that, we'll go to the next slide and I'll turn it back over to Tom.
Thanks, Jeff. So Jeff mentioned the scientific benefits of mapping the pharmacome. This is a little more of an attempt to discuss about the social benefits of mapping the pharmacome. And particularly first, it's a kind of an idea of proactive pharmacology, where we can help avoid the next kind of terrible side effects from certain drugs like thalidomide, fen-phen, or the polyfluoroalkyl substances, which are also known as the forever chemicals, where we can establish pharmacome scale screenings to practically realize and empower scientists to proactively achieve comprehensive understandings of the pharmacology of new compounds introduced in the human environment that can lead to fewer surprises. Secondly, is to be paying it forward. Eve will establish what we hope is a norm for tool sharing. We're going to be generating hundreds of validated reporter cell lines that we plan to make available into the public domain by ATCC or other. If this is a widely adopted technique, as we hope for will be for that can help dramatically reduce new generation pharma mappers costs of entry. And finally, hoping that what we can also help do is to achieve drug discovery in a better, faster, and cheaper manner. In this case, we can enhance the insight into the structural basis of off-target actions and can promote the development of drugs with fewer side effects, hopefully help power drug repurposing for new indications, and establish a public domain pharmacome map, which will make nonprofit drug discovery efforts more competitive and affordable with for-profit pharma. And finally, one question people may have is, so why don't we have this knowledge already? Well, it's kind of threefold. One is that what we are proposing to do is on a scale and cost that would be beyond traditional academic efforts. And in particular, government granting really won't fund it. Secondly, venture capital really doesn't have the time to do this. They tend to more focus on developing new drugs and monetizing them as opposed to studying existing drugs. And finally, large pharma really doesn't have the incentive. One is that they really probably do not want to discover side effects in their own drugs. And secondly, the database that we are proposing to generate, if they were to generate it, it would stay private and be proprietary for their use only. And finally, kind of as a review of the transformative outcomes, we plan on creating a unified, reliable, and searchable database that will illuminate previously unreported positive, negative drug target interactions, which will also help to seed new and or rejuvenate pre-existing drug discovery efforts. And as Jeff mentioned, hopefully provide an unprecedented data set for artificial intelligence and machine learning for drug discovery. And finally, would be to promote the development of new assays, platform technologies, and use for robotics to accelerate drug discovery. And finally, so who we are, William Busa, who is our who is our CEO, who has over 30 years in biotechnology and drug discovery. Myself as the Chief Operating Officer, Jeff DiBerto, who is the Director of Assay Development, a very key component of what we're going to be doing. And Aaron Getz, who also has over 30 years experience in high throughput drug discovery efforts, and Lane McPay as our Director of Data Science. And finally, we are hiring. So if you're interested in what we are doing and becoming a part of it, please reach out to us and let us know. And with that, that is the end of our presentation.